In this video, I will explain a little bit more about high throughput sequencing, read quality, how you read the plots, how you interpret them, and then what you do after that, which is the trimming step. So why you need trimming. So basically, read quality plots indicate what base position in the high throughput sequencing reads start to see a drop off in quality. So I have on the right here an image which looks slightly different from what we look at in DNA Subway, but I think it illustrates some good points. So just like what we look at in DNA Subway, we have forward reads and reverse reads. The y-axis is a quality score, with a higher quality score meaning higher quality sequencing read that we are more confident in, and then the x-axis is the position of that sequencing read in base pairs. So usually sequencing reads are about 250 or less base pairs long. What these quality plots do is summarize all of your sequencing data from all of your samples and then plot for you at each base pair position what overall the read quality looks like for that particular base pair. And in general, like we see in our DNA Subway reads, or like you will see if you haven't done it yet, the read quality generally drops off towards the end of the sequencing read. And so that's to be expected, but really what we can look at in these plots are where we find sequencing reads whose quality is unacceptable. And if you look in these plots, what I like about these is it's color-coded for us in terms of sequencing reads that are really good. So really high quality is in green, the okay quality is in yellow, and then the really bad is in red, which is below 20. And so 20 is the cutoff that I use in our lab this week, and these particular plots are box plots. So if you remember a box plot, what that means is the bottom of the box indicates the first quartile. So essentially, above that bottom of the box is 75% of our data. And so the cutoff that I'm using for our lab this week is to find where the bottom of the sequencing quality boxes gets below 20. That's basically too low of quality, and we don't want to use those reads. But anything above 20, we would consider acceptable. And as I've alluded to a little bit already, read trimming is required because we want to retain only high quality data we're confident in. So at this point, let me mute my computer so you don't have to listen to all my emails pinging in. When we are looking at our sequencing reads, at this point, we're using every single read we've obtained for our data analysis. But really what the quality plots are telling us is that even though the sequencer gave us nucleotides for every one of those 250 base pair positions, the nucleotides that it gave us at the end of the read, it's not very confident in. So a lower quality score means we're less confident that the nucleotide that was assigned is actually the real nucleotide that was present in the sample. And so that's why we have to do this trimming step. We only want to use the high quality reads that we're confident in. So this is an example from our actual data set that we are working with garter snakes. And so this is for the forward read. And if we look here, this is where we start to see a drop off in quality. So here, the bottom of our box plot, the box part of the box plot, or the first quartile, is all the way down like 15 quality score. And so I would say to use this base pair here, 242, as the last good one we want to use. So that bottom of the box is well above that 20 score that we're considering minimally acceptable. And then for the reverse reads, Here, the quality is a little bit lower. So I would say this one here is pretty bad quality. The bottom first quartile goes all the way down below 15. So I would say this one here, 237, is really the last good quality nucleotide we want to keep. And then we trim off everything after that. And for this study in particular, it's a metagenomic study. And as a reminder, what that means is we are taking DNA samples from a community. So in this case, we are looking at samples taken from garter snakes. They swabbed the bacterial community of the garter snakes. And then what we're trying to do is figure out what the bacterial species were in that community. And so for a metagenomic study, we need high quality sequencing reads in order to have high confidence that these species we're identifying in that community are correct. So this is a nucleotide alignment that I've showed in some of our earlier lecture videos. And remember, each line is from a different species or a different taxa, and the asterisks indicate where all the nucleotides are the same. And if we look here towards the end of this particular stretch of sequences, we see that there are several spots where there are actually differences between the species. So at this last nucleotide spot, for instance, this particular species has a G, the other ones don't. So if we were using low quality sequence reads to identify these different species, a base pair mistake at the end of these reads could actually result in us misassigning and saying that it's the incorrect taxa, which is what we don't want. So that's how we need to trim off those low quality reads so we're confident that the species we're identifying in our metagenomic samples are actually correct. And again, just to reiterate that, with the garter snakes, we're taking samples from females and males, and we're trying to figure out what the different bacterial species are in their microbiome community. And so again, we're only using our high quality sequence reads because we want to be confident that the particular bacterial taxa we're saying that are in males and are in females are correct. So that's why we do that read trimming step.